Hi everyone, this is Sadia and today I'm going to explain a presentation that I made about a research article that is named Gut Microbiota's Causal Effects on the Risk of Periodontitis. And it's using a two sample Mendelian randomization study. So, first of all, I will tell you why I chose this study. Because I am a dentist by training and I'm very interested in microbiology related to oral health problems and the dental problems. So, this study is showing us the causal effects of microbiota and that are causing the risk factors, different risk factor of periodontitis. So, first of all, I will explain you the term periodontitis. Okay, so we can divide it into three parts. Perio, first part is perio, that means around, and don means related to tooth, don, and itis means everyone knows it's inflammation. So it's the inflammation around the gums. Inflammation of the gums is the actually the periodontitis. It's a very simple definition that I'm going to explain here. And so the periodontal effects, periodontal diseases are the most common cause of the tooth loss among adults. Okay, and it's a world like most prevalent disease in the world. That in the worst case scenario, it can lead to tooth loss. Okay. Also, it can be related with the systemic health problems like cardiovascular diseases and chronic lower respiratory disorders and diabetes and other like heart related diseases like endocarditis, and it can even lead to the life threatening disease that is Ludwig's angina. So it's a very like prevalent disease, and we should think about the control of the periodontitis. Okay, so here are the some risk factors. Over here, risk factors include the smoking, alcohol consumption, poor oral hygiene, and on so many others. Here you can see the diagram of the periodontitis on the right. It shows that the gums are loose and inflamed, and so there is bone loss and the teeth becomes loose in this condition. Now here is an X-ray, and here you can see the bone loss due to periodontitis, and this is the normal bone level. Now, so the questions that are being asked in this research article is that can gut bacteria affect the gum disease or the periodontitis being connected to the oral cavity? So they are telling the relationship between the gut microbiomes and the periodontitis that happens in the oral cavity. Okay, the other is gut microbes can be seen to be involved with many pathological issues like heart disease, autoimmune, and even cancer. So, can it also cause or affect the periodontitis? So, we already know about the other diseases that can that can cause by the gut microbiomes, but here it's very interesting that they are going to research up, for the periodontitis. And here you can see in the diagram that the oral cavity here is connected to the stomach. So it's all our GIT. So the gut microbiomes are connected to the oral microbiomes in some way, and we will study over here. Okay, so here we have another bottom line answer to the question is researchers use MR analysis that is Mendelian randomization analysis, and GWAS, that is genome wide association studies, to answer these questions. They found a definite causal relation of the gut taxa and periodontitis in this study, and also have expanded the taxa that cause periodontal disease. Okay, so what is the urgency of the question? So this is an issue affecting millions of people and is one of the biggest reasons for the tooth loss, the periodontitis. So having a better understanding and better methods of detecting and predicting periodontal disease is very important, and that's why we have to discover that why is it causing, and why it's causing a lot, and it's getting more and more common among the people, and we have to stop it and control it by some method. So the broader implication to develop more comprehensive treatment for the gum disease. Okay, so now I will tell you about the gut microbiota. As we all know that gut microbiota includes like bacteria, fungi, viruses, archaea, and other organisms. Okay, so there are like millions and trillions of microbiomes in our gut and in our body. So the what are the effects? So huge impact on it has a huge impact on the human health and disease, including immune system, because like uh, they helps in promoting innate and adaptive immunity, and also in digestion, like it helps in digestion of food, cellulose, and also it synthesizes like essential vitamins in our body, and it metabolizes food, and it also helps in our moods and behaviors and neurological diseases. Even the microbiomes can cause cancer. So because they cause inflammation in our gut, so it's a very high risk of for causing cancer. Okay. So the risk factors includes the diet, antibiotics, age, diabetes, and so on. Okay, so now I will tell you about the study, like by which study they are going to find out this causal relationship between the periodontitis and the gut microbiome. So here is it is a Mendelian randomization study, that we can say MR study, and MR study is like a study that is used in epidemiology, and it helps in evaluation of causal relationship between exposure and the disease outcome. It is used to measure variation in genes like um, SNPs, single nucleotide polymorphism, okay, as instrumental variable. Also, I found out that it avoids some of the limitations that we can see, like in observational studies, like reverse causality and confounded. So as I told you about the SNPs over here, so, SNPs is actually a genetic variation. It means like um, single nucleotide polymorphism. Okay, and the SNPs is like uh, in this study we are going to study about the SNPs. Okay, yeah. Okay, so Mendelian randomization study also avoid reverse causality, and as I told you, it has a strong potential confounders. Okay, so here is a diagram that you can see. It has A, B, C. So it's like assumptions, three assumptions that we are going to explain over here, and it's the genetic. Here you can see the genetic variant is connected to the exposure. So it says like the first assumption should be in Mendelian randomization study that genetic variant should be associated with the exposure. And the second one is over here, and it says the genetic variants should not associate with the confounders. Okay, like over here you can see the cross should not associate with the confounders. So confounders are not a good things. And here in the C, the third assumption is the genetic variant should influence the outcome only through the exposure. Here you can see that it should only through the exposure, not the confounders. Okay, so these are three assumptions for the Mendelian randomization study. Now, here is another thing about the MR that it is has some similarities with the randomized control trial. I think you have studied about this trial before. Um, so it is the random MR is similar to the RCT, but uh, because it's similar because the random segregation of the genes separate them independently into exposed and controlled groups. Okay, so that forms unmeasured confounding factors that are also distributed equally between the two groups. So here you can see the exposure and control group. Okay, and here is the same thing, exposure and the control group, so they are similar. Okay, but RCT is considered the gold standard because it is the best possibility to establish a relationship between a risk factor and an outcome. But it has some like disadvantages because the randomized control trial is very costly, impractical, and unethical. So here we are going to use the Mendelian randomization study. Okay, here we will see that they are using that two sample MR study. So what is two sample? So here you can see in the diagram, it's one sample, one sample, one. If we are using only one sample, it's one sample study. And if we are going to using two samples, two different samples, so it's two sample study. So in two sample study uses two different study samples for the risk factors and the outcome to estimate the causal effect of the risk factors on the outcome. So it's helpful when it is difficult to measure the both the risk factor and the outcome at the same time. It also offers the chance to incorporate data from different sources. So whenever the genotype exposure and the genotype outcome data comes from different sources, meta-analysis can estimate the combined effects by using the two-sample MR study to estimate the size of exposure, that is risk factor, and the outcome or of the disease. Okay, so here is association between the genetic instrumental variables. Okay, genetic instrumental variable like SNPs, and the genetic instrumental variables and outcome are generated from two different samples. Okay, here you can see. Then methodology, we will move on to it. We use the MR study in this method, and we found out the causal effects between 196 gut microbiota taxa and the periodontitis in this study. Okay, so the genetic variants we used is are the instrumental variables that should associate with the exposure only, as I explained above. That these were the assumptions, and so the strong instrument variables were established. Okay, here's a diagram that you can see. So uh, here on the left side you can see the healthy state of the tooth, and on the right side here is the periodontitis related uh, tooth and the condition. So here in this method they also here like you can see like um, the green part, that means these are healthy. And there you can see two bacteria microbiota over here, the British coccus and the Rumini clostridium 6, that are related to um, that they are negative. We will I will tell you about it ahead, that they are negative and they don't cause the periodontitis. But over here you can see all these microbiota are causing periodontitis. Okay. And like Probatella, Lactospiraceae, Enterobacteriales, Pasturellas, Enterobacteriaceae, Pasturelaceae, Bacteroideals, Alistipus, and Escherichia. Okay, here is the data collection. Okay, my biogen analyzed genome-wide association genotype and microbiome data from 18,340 individuals from different countries. Okay, so they collected these to study the genome-wide association studies microbiome. Okay, so that included the 196 known bacterial taxa. That includes 9 phyla, 16 classes, 20 orders, 32 families, and 119 genera. So why did they do this? To identify small variation in the genome called SNPs. So SNPs is, as I told you before, that it's helps in identifying the small variation. Okay. I can tell you like about the like because the genome-wide association studies examine SNPs across the genome, they represent a promising way to study complex, common disease in which many genetic variation contribute to person's risk. So SNPs are like genetic variations. Okay, so they are said to be associated with the disease, and they can help researchers pinpoint genes they are likely involved in the disease development. So it's just a variation in the genes that the researchers find out that what genes are genetically variant, and to know the effect of the gene. And do you know that the SNPs have been discovered to contribute to the development of the disease like cancers and more complex diseases? So it's a very good like thing that we can research on it. Okay, so it helps over here we can tell about the GWAS, that it helps to identify genes that are associated with particular disease, study entire set of DNA of a large population, and identify small variations in the genome. Okay, so each study can search for thousands of SNPs at the same time. Okay, so here we can see the minor allele frequency is more than 0.05, and SNP wise call filtering is more than 0.95. Okay, the minor allele frequency is actually is the frequency of minor or recessive gene in a given population. Okay, and it also minor allele frequency helps to differentiate the common and rare SNPs in a population. So it's very good to find out the SNPs. Okay, data analysis for MR study, randomized, random effect inverse variance, IVW, and WM, weighted median, and MR agar. These three things that we will study in this MR study. analysis. And so the it all these will help us to find out the heterogeneity. Okay, so here they used inverse variant weighted as main analysis. Okay, so it was the most robust analysis that provided estimates with heterogeneity. And the other two, that is WM, weighted median, and MR agar, they are just to verify and they prove the validity of the IVW study. That whatever results we found out in the IVW, and they prove the weighted median and MR agar prove this. Okay. So each of these analysis has an odd ratio, confidence interval per standard deviation, and p-values. Okay, so p-value significant was set like less than 0.05 was a significant, and the significant estimate provided by IVW, that is inverse variance weighted, with the same direction of estimates provided by WM and MR agar, was treated as a significant estimate. Okay. So we have more analysis that are called the sensitive analysis and detection of pleiotrope. Okay, so here is the leave one out analysis, MR agar intercept, Cochrane's Q test, and funnel pops. Okay, so the this one is the these all tests were used to detect and confirm the sensitivity analysis. Okay, and pleiotropy. Okay, so pleiotropy, I will tell you about the pleiotropy. Here, the pleo means like many, and tropy means affecting. So genes that affect the multiple genes, multiple apparently unrelated phenotypes or characteristics. Okay, so all these tests over here, they are used to make sure that there is no bias of pleiotropic analysis. Okay, so they were just performed to uh, if there is some heterogeneity. So the first one is the leave one out analysis over here. It was performed to know either the causal estimates was driven by single SNPs, and it is the meta analysis on each subset of studies obtained by leaving out exactly one study. Okay, and the other, the Cochrane's Q test, is also a traditional test for heterogeneity in meta-analysis. Okay, so like we saw in a diagram as well over here, that they perform MR analysis and sensitivity analysis. Okay, so the instrument variables is related to the exposure, not the confounders, and then exposure is goes to the through the exposure they go to the outcome or the result, that is periodontitis, and then they are going to do the MR analysis and the sensitivity analysis. Okay, so now we will move on to our results. Okay, so here are the results that are obtained from the MR analysis. Okay, so here you can see that they use the IWY, inverse variance weighted, WM and MR agar estimates. Okay, so here in the diagram it shows everything, like that you can see a circle, and you can divide this circle in three parts, that is the outer circle, middle, and the innermost. So the outer circle here represents the outer circle shows the IVW value, that is inverse variance weighted, and the middle one here shows the WM, that is weighted median. Okay, values, and the innermost the MR agar. So The outside of the circle there are so many microbiota. There are 196 gut microbiota that are classified into order, phylum, class, family, genus, and also you can see the p-value over here, and that represent different colors. Okay, so of 196 gut microbiota taxa, only 11 gut microbiota taxa associated with periodontitis. Okay, so and out of 11 microbiota taxa, only 7 microbiota show potential positive causal effects. That means uh, they were increasing the risk of periodontitis, and 2 of them were negative effects, and that means they were decreasing the periodontitis and they had negative effect and they were good. Now the we will study some of the microbiota that they found and these results. First of all, Provetella and Lacnis spiraceae. Okay, so they were positively associated with periodontitis, but the problem was their IVW, WM, MR agar analysis were all that were all indicated the positive relationship with periodontitis, but the MR agar analysis exhibit the insignificant trends. But because their p-value was more than 0.05, then that's why they considered it as a positively associated periodontitis causing bacteria, microbiota. Okay, here you can see another diagram for the results that we obtained, and these are the microbiotas over here, and you can see the odd ratios and the p-values. So this diagram is like shows the inverse variance weighted estimates of significantly periodontitis associated with the gut microbiota taxa, and here are 11 microbiota shows in these plots. Okay, these over here. Okay, that were causing some effect on the periodontitis. So the red dots you can see here, they are like represents the IVW estimates, and the black bars over here represent the 95% confidence intervals of IVW estimates. Okay, and it shows the clear odd ratios and the p-values as well over here. Okay, so the microbiota who shows the odd ratio more than 1 indicates the risk of periodontitis, which are 9 over here, like these more than 1, these are 9, and the two of them you can see over here, they are has less than 1 odd ratio. That means they have negative effect or decreasing the risk of periodontitis. Okay, so these two like butyrococcus and Germany Clostridium 6, they have negative effects, and all these 7 have more than 1, so they are have positive effects. Okay, so here we have the microbiotas that we see over here that are causing the increase the risk of periodontitis. Okay, so all these causing the increased risk of periodontitis, and they have the positive causal effects on the risk of periodontitis. Like first of all is the enterobacterials, and so uh, these enterobacterials, as you can see here, is the odd ratio is 1.37, and the 95% confidence interval is like 1.07 to 1.74, and the p-value is 0.012. Okay, so but for the antibacterials and the alistips, these two but their MR agar values are reversed than the other two analysis. They found it out, but they are not significant because due to the absence of heterogeneity of pleotropy and their p-values are more than 0.05. So inverse variance weighted, that is WVM, are more accurate than MR again analysis. So they confirmed and increased that they are increasing the risk of periodontitis. Okay. The others like pastorals, and you can see it has the odd ratio of 1.1 to 95%, CI is 1.01 to 1.25, and the p-value is 0.039. The Enterobacteriaceae is also the order has also the causing the positive effect, and its odd ratio is 1.37, and the 95% confidence interval is 1.07 to 1.74, and the p-value is 0.012. Pastoraliaceae is 1.12, odd ratio, and 95% CI is 1.01 to 1.25, and p-value is 0.039. Okay, so the other two, as I told you, the two on the negative side has the decreasing risk of periodontitis. Okay, these are Butyricoccus and Rumini Clostridium 6, and they have less than 1 odd ratio, as you can see here. Like Butyricaceae has less than 1.082, and the Romani Clostridium is also less than 1, is 0.86 odd ratio. Okay, so here in the results, they after all these analysis, that is they did, and as I told you about that, that IVW, WM, MR, all these evaluations, and they after MR they did the sensitive analysis and the pleiotropy. To minimize the bias effects, these tests were performed and showed the following results. Okay, so leave one out analysis was the first one, and it proved that the 11 taxa do not have pleiotropies in the instrumental variables. Okay, so it proved that no pleiotropy. Then the funnel plots indicated that the no bias being caused by SNPs. Okay, so that there is no genetic variation, no SNP single nucleotide polymorphism. Okay, so no bias. It proved it. And then Cochrane's Q test also showed the lack of heterogeneity. Okay, so it proved our hypothesis as well. The other is MR agar intercept. It also showed that there is no horizontal pleiotropy and among all the 11 microbiota taxa. So they all prove, and the p-value was more than 0.05 of all the 11 gut microbiota taxa. Okay, so it proved that like there is a strong relationship between the instrumental variables and the bacterial microbiota taxa. Okay, and there is no imperfections. Now we will move on to our discussion part. Okay, so as we told about the like all these studies, and they are causing, there are few microbiota that are causing the positive effect, and they are causally initiate the periodontitis, like genus Probertala 7, genus Lachnespiraceae, UCG 008, and order Enterobacteriales, order Pasteurellales, and family Enterobacteriaceae, family Pasteurellacea, family Bacteroideales, S24.7 group, genus Elastips, and genus Esnenbergeria. Okay, and the other two, the genus Butyricoccus, genus Clostridium 6, they had negative effect and causally reducing the periodontitis. Okay, so the Provotella 7, they found it in the fecal samples, and it was the key factor, okay, in the bacteria like Provotella gingivalis. Okay, so like it was inducing the Provotella gingivalis, and it's this bacteria is known for its role in periodontal diseases like periodontitis. So in this study, a higher amount of Provotella 7, like we concluded that it is a predictive biomarker and can be used as target for treatments of periodontal diseases. And the other one is genus Lachnospiraceae, UCG. It also helps in increasing the risk of periodontitis, and so it was among the positive influencing microbiota. And also we found out that they cause some inflammation by producing inflammatory factors like interleukin 6 and tumor necrosis factor alpha, and other some other inflammatory factors that they produce and cause inflammation. And we know that the periodontitis is an inflammation of gums. Okay, so here the order Pasteurellales and the family Pasteurellacea increases the risk of periodontitis as well, and they found in both the oral mucosa and the gut microbiota. Okay, and the family bacteroideals, they also play an important role in the periodontitis, and they cause inflammation. Okay, the other two, as we discussed, that they are decreasing the risk of periodontitis. There is genus Butyricoccus and Clostridium 6. So we also found out that these two actually produce a substance that is butyrate, which acts as an anti-inflammatory mediators. So they inhibits the inflammation, and that means they are not causing the periodontitis because that is inflammation. So they are actually cutting off the inflammation, and they regulate the immunity, maintain intestinal epithelial barrier, balance the gut microbiota, and inhibits the cytokines, because the cytokines are the like the source of inflammation. So they inhibits the cytokine, and they inhibits the inflammation as well. So there are so many targeting number of microbiotas that can help control and prevent the periodontitis. So we know which microbiota are causing and the periodontitis, and which are helping us to inhibit the periodontitis. So we can like invent some methods to prevent and control the periodontitis. And here, as like we did the MR analysis, and it was a very good study. It was very suitable study to find out the causal effects between the gut microbiome and the potential risk factors of the periodontitis. So it was a perfect study, and also it has some advantages like there was no interference of reversal causal effects and confounding factors. Okay, so it's very good that there were no confounding factors and no reverse causal effects in this study. Also, it was very cost effective as compared to the randomized control trial. Okay, And also it was very practical and convincing. So it was the best study for find out the risk factor of periodontitis that are causing by the gut microbiome. But it had some limitations. Okay, so here are the limitations. First of all, GWAS, that was genome wide association studies, was conducted primarily in the Europe, like in the European population. So it can be conducted with other ethnic groups as well. So that was the first limitation. The second one is they didn't focus on the multiple correlations, as periodontitis has so many risk factors and there could be so many reasons. And the third is it had unidirectional role of 196 microbiota. Okay, because they aim to explain the risk of periodontitis only, and they didn't influence on any other role played by these microbiota. So it was only uh, one directional, that is towards the periodontitis. And the fourth one is no explanation of exact uh, exact mechanism of gut microbiota, because they didn't explain too much in detail about these, that how they are going to work and how do they affect the periodontitis. So these were some limitations. Okay, conclusion. So let's move on to the conclusion. So to invent the new techniques for the clinical intervention of the periodontitis, this article explains the causal relationship between the gut microbiota and the periodontitis, that is inflammation of gums, using two sample Mendelian randomization study. So it's very good and like to develop the new techniques to prevent the periodontitis and to control it. Okay, so, questions. First one is, have you ever studied any research that is conducted by using this technique? That is Mendelian randomization. Um, because this is the first time I studied this about Mendelian randomization. And the second one is, do you know about any of these gut microbiota that are related to any oral diseases? Okay, like other than periodontitis, do you know any other oral diseases that are related to gut microbiota? And the third is the two gut microbiota, that is enterobacterials and the enterobacteriaceae, showed reverse effect of the detected values of other two MR analysis. Okay, so why MR agar values are not significant in case of enterobacterials and enterobacteriaceae, and they are still considered an increasing risk of periodontitis. So I just want to know, like, because they had the p-value of more than 0.05. So what do you guys think? Why, if their MR agar values were not supported, so why did they consider it and the positive causing microbiotas towards periodontitis? Okay, so the first question is, what do you think about performing these tests like IBW, WM, and MR agar, Cochrane's Q test, leave one out analysis? So, have you ever used any of these in your research studies? So we can learn more about these tests. Okay, the fifth is, what could be more risk factors of periodontitis and how we can control it? Like other than this, we are going to focus the gut microbiota. Do you know any of the studies that are conducted on the periodontitis for controlling and preventing this disease? And the sixth is, what is genome-wide association studies? And do you know any other study that helped in like gene detection, like genetic variants, SNPs, etc.? The seventh is, what do you know about instrumental variables and confounders? Uh, okay, that's the reference. Thank you.